So I'm going to present today our latest work on 3D tissue maps, unlocking 3D context in spatial biology through interoperable and performant data formats and visual exploration tools. I know it's a mouthful, but I'm going to lead you in. So first question, like why are we actually looking at 3D data, right? We know it's so challenging, it's very costly to produce them, and then like the analysis is very hard as well. And I want to refer to this Nature editorial piece where they say tissue histology is like inherently 3D, and therefore we should also like understand functions and dysfunctions of organs, tissue, functional tissue units, and cells in a 3D spatial context. And there's a really cool paper that just came out from our collaborators, the Sorga Lab, and especially Clarence Yap, the first author here, where they talk about highly multiplex 3D profiling of cell states. And like, because we work so closely together with them, we also heard for them it was very tricky to convince the reviewers, like, what is actually the value that 3D brings. But I think now with this paper being out in Nature Methods, you can really nicely see what you're missing when you don't look 3D. And I want to just very briefly show this video that Clarence created of the data, because like he put a lot of effort into it to really show the breadthness of the data that they have created, and all those like, cells that you can see now in this vast like kind of landscapes of tissue. And of course, such a video is like really cool and nice to look at, but to make sense of the data, it's really tricky. It's really more, like for outreach and getting a sense of like what could we actually perceive and see in this data set. But analyzing it is a different story. Another data set in this direction here is the data set from the Helmholtz University in Munich. And what they were able to is like create a transparent mouse. So this is something you need to do to tissue in order to get to a light sheet microscopy image. So you need to get the tissue transparent, and then you have this light sheet microscope that gets 3D out of the box without having to section the specimen you're looking at. And the really cool thing is they were able to make a whole mouse, of course, a dead mouse, transparent. And now you can investigate like the whole architecture of the tissue in the mouse, like all the vessels and ducts and so on. And they also create really nice videos about these things. But it's, again, very challenging for them to provide this data set to be explored, for example, also in the web, and for other researchers to actually go in and make sense of the data that is being provided here from this group. And so this is kind of a theme that we saw over and over. You can create nice figures and maybe nice videos, but it's very hard to, interactively explore these things and provide it to people. So that brings me kind of like to the challenges that scientists are facing. First, of course, like generating the data is costly and time consuming, but this might get better over time because microscopy technologies also, get better. There's very limited access to viewing software. So like one of the big ones is Imaris, but those licenses are very costly and you need a powerful machine to use it. And of course, then analysis is mainly focused on what one can see, right? So now we're getting these data sets, people look at them. And when we, for example, talk to Peter Sorger, they say like in their lab, they kind of do the eyeballatron. So you kind of like, open the data and zoom and pan till you find something interesting, and then you describe it and talk about it. But that's, of course, not very like reproducible and very hard to understand how you actually got to your findings. And finally, there's a lot of data, but limited insights. And it's also hard to share the data. So even like, if this paper that I just shared is published, it might still be tricky to get access to the data. It's like 1.5 terabytes sitting somewhere in an Imaris file that's like not really downloadable or like viewable for people. So that's kind of like where our group comes in and the efforts that Niels, of course, is leading. And so we kind of try to democratize this access. And one of the first steps there was the VUES library, where there's this cool Avivator website. And it builds upon pyramidal data formats like OMITIF or the newest version, OMI and GFF. And what this allows us to really like pull data on demand into the web browser on the client. So you can kind of view like arbitrarily large data sets on your browser without having an intermediate server between the data source and your client, which are we opening up this website. And so this work was primarily focused on 2D images. So it's very similar to how you would view the world in Google Maps, right? So you can zoom in and out and it loads new tiles and you get better resolution when you close. When you zoom out, you get a nice overview. And now we're extending this into 3D as well. based upon this width that you see up there, we then like developed the VUES platform where Mark is the lead and he's here in the call. And so VUES builds for the spatial data upon width, but then extends it with the capabilities of also showing all like the associated data and experiments that you might have carried out with the data, and then you can view them together, and everything is linked and very nicely set up. And VUES is very powerful in what you can configure it. And Mark will tell us how, like now, we can use it a little easier with easy VUES. So beyond the 2D and what we've just heard, like how can we actually interact with volumes, right? This is like a tricky question. And in volumetric data, we also want to be able to like, select, filter, and sort entities. We want to find the most suitable representation forms. So 3D comes with the problem that you have a lot of occlusion. That means some structures are maybe in front of the structures you actually want to see. And so there are clever ways to work around that. We want to make meaningful selections, which is also tricky in 3D. And so this is one example here, how you could, for example, dissect volumes. So you have like individual views of structures. But what we wanted to like get into is as well to look if these new XR goggles with stereoscopic view, so each eye gets its own display, can actually help to remedy a lot of these problems that we're facing. And the cool thing that these devices bring is they have hand tracking. So we actually go into this room of having the output space and the manipulation space in the same 3D environment. And that's what we investigated here with our paper VUES link. It's a preprint out on arXiv and currently under review. And so what we propose is a hybrid environment where you have the 3D data in your working environment next to your typical 2D desktop environment, and everything is synchronized. So what this allows is you pull up your 3D data in VUES on your desktop, and then you sync it up to your XR goggles with like a 4-digit code. It's very easy, like minimal friction. And then you can really view your 3D data in natural 3D. It's hard to get the benefit out from a 2D desktop environment. So invite, like if you find us somewhere, like try it out together with us, or it's also out there. So if you have goggles, you can give it a shot. And what we got as a feedback from our collaborators is that now, like in 3D, you can see all those like intricate structures way better. Like you see this green thing that was in the middle of the data set is a blood vessel. Sorry, just go back to the video. Is a blood vessel, and it's kind of weaving through the collagen fibers here. And that's very hard to see in 2D, but as soon as you have those goggles on in stereoscopic vision, you can see how it like navigates through. And then we also heard that these cellular neighborhoods and cells very close to each other are very easily perceivable in XR, and gives them kind of like the collaborators a hint where they should focus and put their attention for future analysis. And another thing we heard from Peter Sorga was like he looked at it from the side, and then you can very nicely see if their stainings like actually get through the whole tissue and are in depth as well. And all these are much easier to perceive in XR, and our system is like up and running so people can use it to view 3D data. And so in the beginning, I said, people, like they're limited to just viewing the data and get the sense from theirs about how can we actually now like quantify what we see? How can we get into the analysis part? And so what our collaborators did is like, that's the same data set you've just seen. It's like a melanoma data set with 11,000 cells in there. And they managed to actually segment them out. So now we get into this room of actually working with segmentations of cells, and these can then be used to do quantification. But now that we have those, like the question was, what is a good representation form actually? So for volumetric data and the derived data at the end, we have really nice data sets. So data standards, so it's OMI-NGFF or OMI-SAR. And for the derived data, we have N data, mu data, and so on. But for the middle part, actually, our segmentations, we were lacking a data format that is scalable and is like at the same level as these two you see on the outside. So we did a lot of investigation to find a suitable data format here in the middle. And that's what we now call a 3D tissue map, a combination of raw data, segmentations, and derived data. And we found this data format called NeuroGlancer Precomputed, which you can see here on the bottom right. This data format was created by Google as an open source effort for the connectomics field. And they have a lot of neurons that they want to represent. And they represent them using meshes. It's kind of like the equivalent to a polygon in 2D. And so like it's very scalable. It's pyramidal again. So when you zoom out, you get an overview of all those things. And when you zoom in, you get all the intricate details. That's the middle part here. And while we were on it, we also saw that spatial transcriptomics is having similar issues, right? They have like millions or even like billions of those transcript dots, and they were also lacking a data format to store and retrieve that nicely. And Google also had a solution for this. They call NeuralGlance pre-computed annotations. And so in this effort that we call 3D tissue maps, and star our GitHub repo if you're on it already, we call tissue map tools that helps you to create those data sets and then also shows you how to visualize them. We can now represent raw data, as you see here at the top, with the support of spatial data and OMSR, and then the meshes and spatial transcriptomics dots using the NeuralGlance pre-computed. And as we are like very close to the OMI community, we're also pushing to get this into the standards, which are then provided by OMI and supported by a lot of other tools. So how does that look now when you visualize that in VUES? So currently we have those views juxtaposed. So you see at the top is the raw volumetric data rendering that we provide in VUES, and at the bottom you see there's these meshes, and here we use the NeuralGlance viewer. In the future, we're also planning to bring those two together so you can see the segmentations and the raw data in the same view, but that's an ongoing effort. And the cool thing now is that you can really nicely interact with these. So it's 11,000 cells, and we color them by an embedding that you see here on the right. And VUES brings this really cool interactive features that now, you know, like I can select something in my scatter plot, and I will see where those cells are at in my 3D view. And that's something that was just not possible before. If you spin that a little further, we can now also combine that, like I said, with spatial transcriptomic dots. So here you see 100 million transcripts colored by their genes. And now you see like those meshes popping up. And those are like loaded on demand. It's all on AWS, nothing local. And it just runs on my MacBook. Here it's again 10,000 cells. They're colored by their cell types, as you can see on the right. And the transcripts are colored by their genes. And it's all like interactive in VUES. And so we think now, like as we provide this data set, that it really opens up the opportunity to start analyzing these 3D data sets and then view your analysis together with your data in this interactive way in VUES. And like when we talk to the community, this is just something that like was never possible before. Imaris has very limited support for these meshes. And now with this data standard, we really bring a lot of tools and effort to the people. This again is like a view now in XR. So here we can also do something similar. It's currently less scalable. So it's like only smaller data sets, as you can see here, where we have those meshes and 3D data together. And like I said here, the really cool thing is that I now can interact in 3D with my 3D data, right? So you can really just like reach out with your hand and grab some of those entities and select them, or you can also conduct measurements in the 3D space. And that's also something we want to, you know, like extend a little further and see what the scalability is also in XR for some of those analyses that I just talked about. In the end, you know, like all this potentially with collaborators could also lead to advancing patient care. And that was one approach where we really looked at this, you know, like 3D data sets and thought about what can we actually do now with these. And that's also, again, together with the Peter Sorge Lab. And they're interested in like thinking about communication between cells. So you have like cellular pathways where the cells communicate with each other. And in 3D, this is now like much easier and better perceivable. And so we have one approach in this direction, which is called cell to cell, but it was very manual and very labor intensive to get to your insights. And with this, I kind of want to introduce another thing we're actively working on, and Eunice is going to talk a little bit more about that. It's like really thinking about how we can use abstract representations of the spatial data together with the spatial data in VUES to really make sense of these large-scale data sets. And what you can see here is a cellular neighborhood graph. So that's something we're actively approaching now with the 3D data to see how can now abstracted views like this help us guide and navigate through data sets, and then also help us to now introduce a lot more like, you know, statistical approaches from the graph community to tell us something about which cell types are like mostly neighboring with whom, where they're at in the spatial view, and how can this all be perceived together. And that's kind of like the hint to the next talk that Eunice is gonna give. And with that, I would like to thank you all for your attention.
Thank you, Eric, for the overview of these efforts.